This observatory was constructed to enable the public to engage with astronomy and see the world around them. This presentation introduces the audience to the world on a much smaller scale within the cells of living organisms. As far as we know, the planet Earth is unique within the cosmos, in that it accommodates intelligent life forms capable of reproducing themselves. Life on Earth spans a diverse range of forms, including plants, bacteria, and animals. A fundamental aspect of life is that the blueprint for a new organism can be passed from one generation to the next. Remarkably, all of the independent life forms on planet Earth share the use of DNA to carry this information. The building blocks for DNA form a code which can be divided into units called genes. Genes are fundamental to life on Earth. Studying genes requires a change of scale. Rather than using telescopes to magnify distant objects, optics can be rearranged to magnify tiny objects that are very close. The scales studied using telescopes and microscopes are very different, but sometimes what is observed looks similar. Researchers in Dundee, many based in the School of Life Sciences 1 mile from here, have established a reputation for studying fundamental aspects of cellular life and applying this to understand disease. To illustrate this, we will make use of a tiny 1 mm worm normally found in the soil or in compost heaps. This worm, whose scientific name is C. elegans, is made of 2,000 cells. In humans, many more cells, some 100,000 billion, are arranged to form human organs. In the following clip, Sidney Brenner explains why he chose to study the tiny worm C. elegans.
What you want is to have the whole problem preserved in essence. So we need an organism that's got to have lots of different cells, it's got to be etc., etc. And if possible, it's got to have a nervous system. And one was a little round worm that lives in the soil with a life cycle that's very fast, okay? So that you can go through 2.5 day life cycle, means in a week you can do 2 generations, something like that. And we sat down, we worked out how to do genetics with these. We sat down, we started to do electron microscopy, we started to reconstruct the things, and that is the way it grew. We got mutants, and when people say, what are you going to do with the mutants? We say, we're going to study them as deeply as we can.
Despite their small size and simplicity, C. elegans has proven to be useful both for basic research and understanding the basis for human disease. Who would have thought that studying such a primitive organism would have been so informative?